Hello my dear students, welcome back to Biology at Physiology. Today we'll learn about bacterial taxonomy. Bacterial taxonomy deals with the identification, classification and naming of different bacteria based on their morphological, physiological, biochemical, molecular and ecological characteristics. So, modern taxonomy integrates classical methods with molecular and genomic approaches to provide a more accurate understanding of bacterial diversity and evolutionary relationships. Introduction to bacterial taxonomy. Bacterial taxonomy is the science concerned with the identification, classification and nomenclature of bacteria together with the study of their relationships. There are three important words: identification, classification and nomenclature. Identification means determining what an unknown bacteria means. Classification means placing the bacteria into an appropriate taxonomic group based on its characteristics and relationships, and basically nomenclature means assigning the organism a scientifically accepted name according to the appropriate rules. But bacterial taxonomy has evolved considerably over time. In the early days of microbiology, scientists mainly depend on what they could observe, that is cell shape, staining behavior, colony morphology, biochemical reactions and physiological properties. These approaches are still useful today. So, however, modern microbiology has shown us that the phenotypic similarity doesn't necessarily mean evolutionary relatedness. Two bacteria may look almost identical under the microscope but may belong to completely different evolutionary lineages. Therefore, modern bacterial taxonomy follows a polyphagic approach. What is polyphagic approach? Polyphagic approach means that several, poly means many, or phagic means characteristics or phases. So polyphagic means that several different types of evidences are considered together in bacterial taxonomy, such as morphological characteristics, staining characteristics, cultural characteristics, physiological characteristics, biochemical characters, chemotaxonomic characteristics, serological parameters, molecular or genotypic phenomena or features, phylogenetic characteristics, ecological characteristics. So let us understand each of these systematically. What is morphological characteristics? The first character we usually observe is morphology of the bacteria. Morphological characteristics refers to the physiological appearance and structural features of bacterial cells. This is often the starting point for bacterial identification because microscopy is relatively simple and provides immediate information. The most basic feature is bacterial cell shape. Bacteria can be cocci, bacilli, vibrios or spirilla and spirochetes. Uh, but shape alone is not sufficient. We must also consider cellular arrangement. For example, cocci may occur as single, in pairs, diplococcus, in chains, streptococci, or in grape-like clusters, staphylococci. Some may occur in groups of four called tetrads, while others form cubical packets known as sarcini. We can also examine structures such as capsule, flagella, fimbriae or pili and endospores. Endospore formation is particularly useful. Genera such as Bacillus and Clostridium are well known for their ability to produce endospore. Even the position of the endospore, either central or terminal or subterminal, can provide additional information. However, here is an important point for examination. Morphology is useful for preliminary identification, but morphology alone cannot establish bacterial identity or evolutionary relationships. Why? Because unrelated bacteria can independently evolve similar shapes. This is a classical example of why phenotypic similarity is not always equivalent to phylogenetic similarity. Staining characteristics. The next major characteristics is staining behavior. Staining allows us to visualize bacteria more clearly and, more importantly, reveals differences in their cellular structures. The most important differential stain in bacteriology is the Gram stain, which was developed by Hans Christian Gram. Based on this procedure, bacteria are broadly divided into gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria can retain the crystal violet iodine complex stain during the decoloration step because of their characteristics thick peptidoglycan rich cell wall. They therefore appear purple or violet. Gram-negative bacteria have a much thinner peptidoglycan layer and an outer membrane. They lost the primary stain during decolorization and subsequently take up the counter stain, commonly safranin. They therefore appear pink or red, but staining does not stop with the gram stain, and important method is the acid first stain. It is particularly useful for organisms possessing cell walls rich in malic acid, especially members of the genus Mycobacterium. For example, Mycobacterium tuberculosis and Mycobacterium leprae. We can also use special staining techniques to demonstrate capsules, endospores and flagella. Therefore, staining characteristics provide an important bridge between cell structure and bacterial classification. Cultural or colony characteristics. Now imagine that instead of looking at individual bacterial cells, we all allow the bacteria to multiply on an artificial culture medium. After incubation, we may observe visible colonies. The appearance of these colonies is called the cultural or colony morphology. Different bacteria may produce colonies with distinctive size, shape, color, surface texture and margin, elevation, opacity and consistency. For example, Staphylococcus aureus may produce smooth, opaque colonies that can show characteristic golden yellow pigmentation. On the other hand, Pseudomonas Aeruginosa can produce pigments that may give colonies a greenish appearance under suitable conditions. Another important characteristic is hemolysis on blood agar. Bacteria may produce alpha hemolysis or beta hemolysis. Alpha hemolysis is the partial destruction of red blood cells, often producing a greenish zone. Beta hemolysis is the complete hemolysis, producing a clear zone around colonies. And gamma hemolysis is essentially no hemolysis. These observations can be highly useful in diagnostic bacteriology. However, colony morphology is influenced by factors such as culture medium, temperature, incubation time, atmospheric conditions and nutrient availability. Therefore, colony characteristics should be interpreted together with other characteristics. Physiological characters. Now we move from what bacteria look like to how they live. The physiological characters describe the evolutionary and nutritional conditions required for bacterial growth. Temperature is one important factor. Based on temperature preference, bacteria may be broadly classified as cyclophiles, mesophiles and thermophiles and hyperthermophiles. Similarly, bacteria differ in their pH tolerance. So we have three kinds of bacteria: acidophiles, neutrophiles and alkaliphiles. Another highly important physiological characteristics is oxygen requirement. Bacteria may be obligate aerobes, obligate anaerobes, facultative anaerobes and microaerophiles and aerotolerant anaerobes. This information is particularly important because bacterial oxygen availability strongly influences their metabolic processes. Some bacteria also require high concentration of salt and they are referred to as halophile. Others have highly specialized nutritional requirement or possesses adaptations allowing them to survive extreme environmental conditions. Thus, physiological characteristics help us distinguish bacteria that may look similar but have completely different ecological lifestyles. Biochemical characters. Now we come to one of the most important classical approaches to bacterial identification, that is biochemical characterization. Every bacterial species has a characteristic collection of metabolic capabilities. In simple words, different bacteria perform different biochemical reactions. Why? Because bacteria possess different enzymes and metabolic pathways, and these differences can be exploited in the laboratory. Some commonly used biochemical tests include catalase test, oxidase test, urease test, citrate utilization test, indole test and methyl red test, Voeg's Proskauer test, nitrate reduction test. We can also examine whether bacteria can ferment particular carbohydrates, produce gas, hydrolyze proteins, degrade lipids, utilize particular carbon sources. When these results are combined, we obtain what can be described as metabolic fingerprint. One individual biochemical reaction may not be sufficient to distinguish two organisms, but a combination of biochemical reactions can provide a highly useful identification profile. This is why biochemical testing has traditionally been such an important component of clinical and environmental microbiology. Chemotaxonomic characteristics. Now let us move to a more specialized level, that is chemotaxonomy. Chemotaxonomy means classifying organisms using characteristic chemical components of their cells. The basic principle is simple. If a particular chemical component is consistently associated with a particular taxonomic group of bacteria, it can serve as a taxonomic marker. For example, we can say that cell wall composition, including the structure and composition of the peptidoglycan. Other important markers we can include fatty acid methyl ester profile or FEM profile, profile, respiratory quinones, polar membrane lipids, mycolic acids, certain characteristic cell wall sugars and amino acids. These chemical markers can be particularly useful when closely related bacteria need to be differentiated. For example, mycolic acid composition is highly informative in the taxonomy of mycobacteria and related organisms. One classification is important here. DNA G plus C content has historically been used as a genomic taxonomic characteristic rather than a purely chemotaxonomic marker. It can provide supporting information about genomic composition, but by itself it does not establish phylogenetic relatedness. Therefore, modern taxonomy interprets such characteristics along such sequence and genome-based evidence. Serological characters. Next comes serological characterization. Serology is based on the interaction between antigens and antibodies. Bacterial cells possess a variety of surface antigen. Some important examples include O antigen, H antigen and K antigen associated with capsular structures in organisms where such antigenic systems are recognized. These antigenic differences can be used to distinguish different serotypes or serovars. This approach has been particularly important for pathogenic bacteria. For example, different strains of Salmonella, E. coli can be differentiated based on their antigenic profiles. Serological typing is especially valuable in clinical microbiology, disease surveillance, epidemiology, outbreak investigation, etc. Although serology is not the primary basis of modern bacterial phylogeny, it remains valuable for differentiating strains and understanding epidemiological relationships. Molecular or genotypic characteristics. Traditional methods examine what bacteria look like and how they behave, but molecular methods examine their genetic information. So one of the most influential molecular methods in bacterial systematic is the 16S rRNA gene sequencing. DNA hybridization include the other processes, whole genome sequencing, average nucleotide identity, etc. Molecular phylogenetic characteristics. Molecular data allow us to go one step further. We can ask not merely what bacterium is this, but also where does this bacterium fit into evolutionary history of life. This is the basis of phylogenetic classification, such as 16S rRNA gene sequences, conserved protein sequences, whole genome data, comparative genomic information to construct phylogenetic trees. So therefore, morphology tells us what an organism looks like, but phylogeny attempts to tell us whether that organism fits in evolutionary history. Ecological characteristics. Ecological characteristics provide important supporting information for taxonomy, such as some bacteria have remarkable adaptations that enable them to survive extremely high temperature, high salinity, acidic environment, high alkaline conditions, high pressure, etc. So ecological characteristics alone are generally not sufficient to establish bacterial classification. However, they provide valuable supporting information about adaptation, ecological diversity, physiology and evolution. Why can we not depend on a single character? Now let's bring all these characters together. Suppose we find a bacterium that is gram negative, rod shaped, motile and capable of producing particular biochemical reaction. Is that enough to identify the organism? Usually not. Why? Because thousands of bacterial species may share some of these properties. Even a combination of several phenotypic characters may sometimes fail to resolve closely related organisms. This is where the concept of polyphagic taxonomy becomes extremely important. So polyphagic taxonomy means that we do not depend on one type of evidence, instead we integrate in the multiple lines of evidence such as phenotypic data, chemotaxonomic data, genotypic data and phylogenetic data and ecological data. Genome sequencing may provide the detailed picture. When all these pictures agree, our confidence in the taxonomic position becomes much stronger. So conclusion, the more independent evidence we integrate, the more confidently we can determine the identity, classification and evolutionary position of a bacteria, and that is why bacterial taxonomy is not simply the art of naming bacteria. Instead, it is the science of understanding bacterial diversity, identity, relationship, evolution and biological significance. So if you are studying microbiology, this concept of bacterial taxonomy is important, very much important for you. In your next discussion, we can go one step deeper into modern approaches of bacterial taxonomy. So my dear students, connected with biology at Physiological for more concept based lectures in microbiology and other subjects of life science. Thank you for watching. Keep learning, keep questioning and keep exploring the microbial world.